Welcome to the Timeless Science. The human brain treats memories of the past and images of the future using nearly identical neural systems, even though one reflects lived reality and the other pure imagination. This paradox challenges the idea that memory is a reliable record of time. In this episode, you will hear how neuroscience overturned the belief that memory works like a mental archive, and why the brain was never designed to store the past, but to simulate possible futures. For centuries, memory was assumed to function as an internal recording device. Experiences were thought to be stored, preserved, and later retrieved with reasonable accuracy. This assumption felt intuitive, because memories often feel vivid and emotionally grounded. Yet neuroscience began dismantling this view in the late 20th century. In 1985, Endel Tulving at the University of Toronto introduced the concept of episodic memory, defining it as the ability to mentally travel back to specific personal events. Tulving emphasized that remembering is not passive playback. It is an active reconstruction occurring in the present moment, shaped by current goals and expectations. This insight marked a turning point. Memory was no longer treated as storage but as a process. In 1997, Tulving further clarified that episodic memory always involves self-projection. When a person remembers, the brain reconstructs a scene using fragments rather than retrieving a fixed copy. What matters is not accuracy but coherence. The brain fills gaps, smooths inconsistencies, and prioritizes meaning over detail. This explains why confident memories can still be wrong. The neurological basis of this reconstruction became clearer through imaging studies. In 2007, Eleanor Maguire at University College London studied patients with damage to the hippocampus, a structure critical for memory formation. Her team observed that these patients struggled not only to recall past experiences, but also to imagine new ones. When asked to picture hypothetical future events, their descriptions collapsed into vague fragments. Functional MRI scans showed reduced activation in the same networks used during memory recall. This finding was critical. It demonstrated that remembering the past and imagining the future are not separate mental functions. They rely on the same neural architecture. The hippocampus does not store time. It assembles scenes. In 2012, Daniel Shatter at Harvard University formalized this idea as the constructive memory theory. His experiments showed that the brain recombines elements of past experience to simulate future scenarios, using identical patterns of neural activation. The implication is profound. Memory exists not to preserve history, but to enable prediction. The brain reconstructs the past because it uses those reconstructions to anticipate what might come next. Accuracy is secondary. Survival depends on flexibility, not fidelity. This reframes memory as a tool for simulation rather than recollection. With this foundation established, the next chapter will examine how the brain became a biological time simulator, and why imagination and memory are neurologically inseparable. The idea that the brain simulates time rather than stores it emerged when neuroscientists began comparing the neural activity of remembering and imagining. What they found was unexpected. The same large-scale network became active whether a person recalled a childhood event or envisioned a future scenario that had never occurred. This network, known as the default mode network, links the hippocampus with regions of the medial prefrontal cortex and posterior cingulate cortex. Its function is not tied to the present moment, but to internally generated scenes. In 2007, Eleanor Maguire and her team at University College London used functional magnetic resonance imaging to observe brain activity during memory recall and future imagination tasks. Participants were asked either to remember a specific personal event or to imagine a plausible future experience. The scans revealed nearly identical activation patterns. The hippocampus played a central role in both cases. This result demonstrated that the brain does not switch systems when moving from past to future. It reuses the same machinery. The hippocampus acts as a scene constructor. It binds together sensory fragments, spatial layouts, emotional tones, and contextual details into a coherent mental experience. When remembering, it reconstructs a past scene from fragments. When imagining, it recombines those fragments into a novel configuration. In both cases, the process is generative rather than archival. What matters is whether the scene feels internally consistent, not whether it is historically accurate. Further evidence came from clinical observations. Patients with bilateral hippocampal damage could describe facts about their lives, but they could not mentally place themselves in time. In 2008, McGuire documented that such patients failed to imagine detailed future events. Their descriptions lacked depth, spatial structure, and continuity. The brain was not unable to think about the future. It was unable to simulate it. This finding revealed the true function of episodic memory. It exists to support flexible planning. By reconstructing past experiences, the brain extracts patterns and uses them to anticipate possible outcomes. In 2012, Daniel Shatter at Harvard University demonstrated that imagined future events are built from recombined elements of real memories. Functional imaging confirmed overlapping neural signatures. The brain was not confusing past and future. It was using the past as raw material for prediction. The consequence is clear. Time in the brain is not linear. It is a workspace. The same neural system generates memories, plans futures, and constructs hypothetical realities. The brain does not travel through time. It simulates it. The next chapter will examine what happens when this system breaks, and how damaged brains revealed the strongest proof of this paradox. The strongest evidence for the brain's inability to separate remembered time from imagined time emerged when the system itself was damaged. Clinical neurology provided conditions that laboratory tasks could not. When specific brain structures fail, their function becomes visible by absence. In the case of memory and imagination, the collapse was identical. In 2003, researchers at University College London examined patients with selective damage to the hippocampus caused by oxygen deprivation. Eleanor McGuire led the study and documented not only severe autobiographical memory loss, but an unexpected secondary deficit. These patients could not construct detailed future scenarios. When asked to imagine ordinary events such as a walk on a beach or a future meeting, their descriptions were brief, fragmented, and spatially empty. The patients were not confused. They were calm and articulate. What was missing was the ability to mentally place themselves inside a scene. This result was not anecdotal. In 2008, McGuire repeated the experiment with controlled prompts and neuroimaging. Functional MRI scans showed that during both memory recall and future imagination, healthy subjects activated the hippocampus along with the medial prefrontal cortex and posterior cingulate cortex. In patients with hippocampal damage, this network failed to engage. The same network collapsed in both tasks. The deficit was not memory-specific. It was simulation-specific. Parallel findings came from the United States. In 2012, Daniel Shatter at Harvard University conducted a series of experiments measuring neural overlap between past recollection and future projection. Using functional imaging and behavioral scoring, his team demonstrated that imagined future events drew directly from recombined elements of stored experiences. When participants were instructed to imagine novel futures, their brains activated memory retrieval circuits first. When those circuits were impaired, imagination degraded. Additional confirmation came from neurological disorders affecting broader networks. In 2010, researchers at the National Institute of Neurological Disorders and Stroke in Maryland studied patients with early-stage Alzheimer's disease. These patients showed parallel declines in autobiographical memory and future planning. As hippocampal degeneration progressed, both abilities deteriorated together. The loss of memory and the loss of imagination were not sequential. They were simultaneous. These observations eliminated alternative explanations. The brain was not confusing past and future due to failure. It was revealing its design. The hippocampus is not a memory vault. It is a scene generator. When it fails, the brain loses access to time itself, regardless of direction. This clinical evidence forces a conclusion. Remembering and imagining are not separate operations that share resources by coincidence. They are the same operation applied to different temporal labels. The final chapter will resolve why this system evolved, and why the brain treats imagined time as real enough to guide action. The paradox can now be resolved by understanding what the brain is optimized to do. The human nervous system did not evolve to preserve the past. It evolved to regulate behavior in an uncertain environment. From a biological perspective, the past has value only insofar as it improves future action. This single constraint explains why the brain treats remembered time and imagined time as functionally equivalent. Neuroscience shows that the brain operates as a predictive system. In 2010, Carl Friston at University College London formalized this idea through the free energy principle, proposing that the brain continuously generates internal models to reduce uncertainty about the world. These models are tested against sensory input and updated over time. Memory is not an archive in this framework. It is a database of components used to build predictions. When the brain recalls an event, it is not asking what happened. It is asking what information from the past can help regulate behavior now. When it imagines the future, it asks the same question. Both processes involve constructing an internal scene detailed enough to evaluate outcomes. The brain does not label one as past and the other as future at the neural level. That distinction exists only at the level of conscious interpretation. This explains why imagined experiences can feel emotionally real. In 2014, researchers at Stanford University demonstrated that imagined future events activate the amygdala and autonomic nervous system in patterns similar to real memories. Heart rate, skin conductance, and emotional tone responded not to reality, but to plausibility. The brain reacted to simulations as if they were meaningful experiences. From an evolutionary standpoint, this makes sense. A system that waits for certainty before responding is slow. A system that simulates outcomes can act in advance. The cost of occasionally reacting to imagined threats is outweighed by the survival advantage of preparation. Accuracy is less important than usefulness. The brain evolved to treat internally generated time as real enough to guide decisions. This reframes human consciousness. The feeling of moving through time is not a passive experience. It is an active construction. The present moment is surrounded by simulations of what has been and what might be. The same neural machinery supports regret, anticipation, planning, and identity. The brain does not store a timeline. It continuously rebuilds one. The paradox dissolves here. The brain cannot distinguish between remembered time and imagined time because, at the level that matters for survival, there is no meaningful difference. Both are simulations designed to control future behavior. Goodbye. Thank you for listening. Perhaps, see you in the next frame of understanding.